All right, so today our sixth seminar for this semester comes to us from Georgia Institute of Technology out there in Atlanta. Our speaker is Professor Stefan France. Maybe I'm saying that wrong.
That's right.
That's right, okay. Okay, so he's a native of the Bronx in New York. Did his BS in chemistry from Duke University, MA and PhD in chemistry from Johns Hopkins, and is a synthetic organic chemist. You guys could guess why I was interested in hearing what he had to say. And is associate professor of chemistry now at Georgia Tech. His areas of interest in terms of research is method development, developing new synthetic methods, natural product synthesis, and medicinal chemistry. Are we offering medicinal chemistry this semester? Or was that last semester? Okay, so some of you have taken that medicinal chemistry class, so this may have some things that may be of interest to you. So Professor France has been a recipient of many different awards, including 2015 Senior Faculty Outstanding Undergraduate Mentor Award, 2014 Faculty Award for Academic Outreach, 2013 Sigma Psi, and 2012 a Nobushi Award. And the NSF Award is actually money, big money. So that's a very good award for anyone to get. So it's an honor and a privilege to have you here with us. We look forward to your presentation.
Thank you very much for the kind introduction. It's my pleasure to be, to present for you guys. This is new for me. I've never actually done a virtual seminar before, so I'm hopefully I do a good job and you guys actually enjoy the seminar. But I definitely thank you for the invitation. And hopefully, by the time the talk is done, you can appreciate some of the stuff that we're trying to do and some of the things we're thinking about in terms of chemistry, in terms of synthesis, in terms of total synthesis, things like that. Okay, and hopefully you guys can hear me fine. There's no issues sound-wise. Okay. So what I wanted to do today is tell you a bit about some of the stuff we're doing, highlighting some of the methods that we've developed, which we then are trying to use for specific application. And what I want to do is I'll give you a little bit of overview about the group, specifically the way we think about some of the chemistry, and then try to highlight 2 or 3 vignettes about some things that we're actively engaged in right now. We'll see how the time goes if we get through all 3, but I want to make sure we keep the time and things like that. So what you'll see is this title slide, you'll see the main figure. That's going to be the heart of this talk, where basically we're looking at the concept of diversity-oriented synthesis, and basically for specific methodology, where we change the methodology, change aspects of the method, where we can access different kinds of compounds, different structures, and things like that. And I'll talk about that more in detail shortly. So we're a synthetic organic group, as was mentioned, where our focus in terms of using the chemistry is in terms of method development, in terms of synthesis, is looking at efficiency, modularity, versatility. Those are the aspects we really look at when we're trying to think about methods, trying to think about how to approach doing a complex synthesis. And the aspect of it means that when we do the synthesis, we can actually put that toward different applications, whether it be for the method itself, for total synthesis, for more biologically relevant processes or protocols, or if we're looking at anything materials or environmentally focused, things like that. So we're actually interested in a wide range of topics, but at the heart of it is all utilizing tools of synthetic organic chemistry to be able to achieve all the goals that we want to achieve. So just a little overview of my lab. So research, methodology development is a good heart of what we do. And we've done some, a bunch of work on ring opening cyclizations. And so we'll talk about some examples of that as we highlight the vignettes. We really are now pushing more towards natural product synthesis with an eye towards targeting molecules that are bioactive, whose activity may not be, super, highly potent. Some of them may be modestly potent, but then trying to develop modular approaches to the method to allow for some structure-activity relationship studies. Especially if the mode of action is not known, can we go towards solving that aspect, or can we actually develop more therapeutically active or potent compounds? We've done a lot of collaboration with other groups towards medicinal chemistry, particularly in the realm of small molecule or semi-synthesis in natural products, where you take a natural product and do just manipulation of that target itself. In the 4 examples I've shown there, we start with the first one here, where we're making these flavone compounds and looking at these compounds as neuroprotective agents. And so we synthesized several derivatives of this with Keishan Yi, who's at Emory. We did some work with them. We teamed with them again, looking at these small molecule derivatives that were for mimicking insulin. And so we had some results with that, teaming with them. This natural product compound is an anti-malarial compound. And we worked with a group that's at Georgia Tech, Julia Kubanek, where she does natural product isolation, and we did some semi-synthesis, and we're able to help her and her group determine the mechanism of action of these anti-malarial compounds. One of the projects that I'm not going to talk about today is we're actually looking at developing a modular synthesis of this family of compounds, because there's actually over 25 members of the family, all with some anti-malarial, some anti-cancer, some antibacterial activity. And so we're looking at developing methods to go after those compounds. And then working with Raquel Lieberman, who's a biochemist here at Georgia Tech, also looking at small molecules, particularly towards glaucoma proteins. She's a protein crystallographer, so we made ligands where she was doing crystallography and developing assays to try and see novel binding patterns and things like that. Okay. Beyond the standard, your medicinal chemistry method develops and developmental synthesis, we're also interested in other areas. So I'm part of a NSF-funded Center for Chemical Evolution, where we look at the self-assembly of small molecules to more complex systems, to how then you can have function, which would basically mimic what we have today in life. We also are looking at the utilization of things like cyclodextrins to do self-assembly for purposes of anything from carbon nanotubes, transport, separations, things like that. We've done some work with the chem engineering group, doing some flow chemistry as we try to synthesize compounds on a more efficient protocol. And then we're also interested in materials, mainly because some of the methods that we're developing allows for the synthesis of conjugated systems. Okay. But that's an overview, and we're interested in a lot of different areas, but the main thing is, at the heart of it, is we can utilize the tools of organic synthesis to be able to access and apply in different areas. Okay, so to start, in terms of what we're really thinking about, is in the title part was the term diversity-oriented synthesis. If you haven't heard that terminology, that term is often represented as DOS or DOS. It's the facile preparation of collections of structurally complex and diverse small molecules from simple starting materials. And so if you take this standard synthetic protocol, standard synthetic protocol where you're doing retrosynthetic analysis, you start from some target molecule, you figure out disconnection approaches, and then look back on how you get to that molecule from simple precursors. That's your classic approach. Diversity-oriented synthesis approach utilizes a divergent pathway where you start with some common precursor, and then you use that precursor to divertize as much as possible, where you get to a point where you have a lot of chemical space that you've just accessed. And so these are complementary approaches, but both allow for the ability to start thinking about, if I have a target of interest, can I develop methodologies that allows for to develop analogs, to develop other compounds that can be readily tested or developed for other purposes. Okay, and so there's a classic example from the Schreiber lab, one of the pioneers of diversity-oriented synthesis, where you're looking at starting from some precursors. And usually when you think of diversity-oriented synthesis, it's this whole bill, couple, pair iterative sequence where you start with different components, you couple those components in some capacities to build up complexity, You pair them in different ways so you can start, branching out further so you can build up more complexity for the system. But what you do is start with simple precursors, and you get more diverse and more complex as you go, down that chain. Another type of synthetic approach that we're also interested in is the idea of taking diversity into synthesis and looking at a mix and match type approach, which you can liken to chemical Legos. Right? And I have a 7-year-old son who's big into Legos, and it's all about taking your imagination, thinking about how you can look at a pile of Legos and then envision building whatever is in your mind. And so that's the way I try to look at organic synthesis: is can we develop methods that allow for a mix and match approach where you can start just changing pieces around and building what you want, just based on taking advantage of the mechanistic understanding of the system. And so that's our framework that we use in my lab, is really simple building blocks and build up as much structural complexity as possible and utilize that to really think about doing a complex synthesis. When we're talking about natural products, our goal is not just the target itself. Our goal is actually make the natural product itself, but being able to make a method that allows for derivatives and analogs to be generated, particularly because that allows us to then look at biological activity. And this terminology is often looked at as diverted total synthesis, where you prepare the natural product, but really what you're doing is preparing a library from some common intermediates, and then you can derivatize them and make as many analogs as possible, with hopes that the various analogs tell you something about the biological activity, tell you something about the mode of action, but also with the idea that can you use that to ultimately develop some therapeutic value or other probe aspects or things like that. Classic example comes from Sam Danishevsky's group, where apotholone B, that natural product, it was found to have very severe toxicity, even though it was fairly potent. And so by changing the nature of the compound itself, so removing the epoxide that decreased toxicity, getting rid of, putting in unsaturation at this position here, and what you started getting was compounds that were more potent. So these compounds here are in phase 2 clinical trials. These compounds here, phase 1. Changing different aspects of the system, you get to compounds that are much more potent, much more diverse, and allows you to really start thinking about starting from a molecule that will never be a drug. Can you actually start putting together some changes in the molecule where it actually now becomes pharmaceutically relevant? Okay, and that's the kind of thing we think about when we're looking at total synthesis. Can we, put together the molecule in a rapid fashion, but allows for rapid assembly of a library of compounds for testing? Okay. So that leads us to talk about some of the methods that we've developed in the lab. Particularly, the method I want to talk about is the catalytic formal Homo-Nazarov cyclization. It's, I'm going to try to keep it as topical in a sense, without getting too nitty gritty into the hardcore details of the methodology and things like that. I just want to highlight what the key aspects of the method are and what you can actually do with it when you start thinking about it in a mix and match type approach, or in a change this component, you get this, change this other component, you get that, and being able to utilize that to, develop and access a lot of chemical space. And so, as a template for diversity synthesis, we look at using strained ring systems, where you have a strained ring system and you have an adjacent pi system that once you do a ring opening, you end up with a dipolar intermediate that is attacked by the adjacent pi system. And what you do is do a ring opening cyclization. So you go from that 3-member ring to a larger ring system, usually either 5, 6, or 7, depending on the type of ring, depending on the type of pi system, and so on and so forth. And so what I'm showing here is different kinds of DOS. When you think of DOS, there's basically changing the periphery of your molecule to get libraries that are just changed in different different portions of the molecule. There's reagent base where you bring in different reagents, and that gives you different products. And then there's substrate base where if you change different components of the molecule, it totally changes the overall framework of the molecule that you get. So you get totally different chemical architectures and chemical structures. And so what I want to highlight today is really, I'm going to take a vignette from these three, the appendage manipulation-based DOS and the substrate-based DOS, and try to highlight some of the things we're thinking about in terms of going, particularly for the first two, we'll highlight some total synthesis, where they were trying to take that and do some medicinal chemistry, but then the substrate-based, we're taking that and trying to do some, make some interesting chemical probes. Okay. So why do we care about using strain ring systems? So cyclopropanes are an interesting class of strain rings. They're pretty unreactive on their own, but if you put the right substituents about the cyclopropane, they become fairly reactive. And so in particular, for cyclopropane, if you put a donor group on one carbon and an acceptor group on another carbon that's adjacent, what you get is a push-pull effect. So that's what this cartoon is showing. The donor group's pushing, the acceptor group's pulling, and what's happening is the bond in between gets polarized, or that's the one that's ultimately going to get broken. So you get some good control over that system. And what that actually means is when you do that, there's different donor groups and acceptor groups you can use for that, but it means that you access a wide range of reactivity, particularly because you go between the regimes of where it's fully ring closed to the cyclopropane to where it's a full charge separated 1,3 dipole. And these systems, they undergo a wide range of reactions, whether it be with electrophiles, nucleophiles, you can do cycloaddition chemistry, they can undergo rearrangements. And so what we're hoping to do is take advantage of this ring opening cyclization, but in an intramolecular fashion to allow for the formation of specific ring sizes and ring systems. When you look at the donor acceptor cyclopropanes, putting more acceptor groups or more donor groups on the system increases or tunes that bond cleavage. And so we were really interested in taking advantage of this fact in designing donor acceptor cyclopropanes to do intramolecular chemistry. There wasn't a whole lot of examples of the intramolecular chemistry of this type, particularly for the kind of reactions we wanted to do, where we want to be able to catalyze them by Lewis acids. And so when looking into the literature, there's the normal mode where you generate your positive charge towards your donor, a negative charge towards your acceptor, and then it reacts with some, electrophile or nucleophile according to that charge. A less common way is switching the polarity, so an up-along approach. There are a few examples of that in literature, but we were really primarily interested in the normal approach. And so the kind of literature you see on these donor acceptor cyclopropanes, particularly with Lewis acids, usually involve the secondary acceptor, where you can react with a Lewis acid and get this binding mode, where that binding mode really polarizes the cyclopropane. It allows for really good control over the reactivity. And so this is the kind of binding mode that we're going to focus on in terms of thinking about the design for our cyclization. And I will say, feel free to ask questions as you're going if there's anything that I can clarify or things like that. So when we were looking at this, our design for our reaction, our inspiration was this, as I mentioned, the Homo-Nazarov cyclization, where it's a cyclopropyl vinyl ketone. Okay, so cyclopropyl vinyl ketone. You treat it with a Lewis protic acid to activate the system. So you activate the carbonyl. You get ring opening of the cyclopropane to give you a 1,3 dipole, and then the pi system attacks that carbocation. So the result is this 6-membered oxyallocation, which then can undergo loss of a proton and then tautomerization to give you a cyclohexanone. So that's the premise of the baseline reaction. The first example of this was shown in the 1980s, but there's only been one example of catalysis, and that was in about 2010. And we were really interested in expanding this reaction because it hadn't really been exploited for any useful synthetic purposes. And so with the model that we had wanted to put in, where we added the secondary acceptor group, this was the modified reaction design that we use going forward, where you basically have now that secondary group. You treat it with a catalytic Lewis acid so that you get that coordination mode, which helps facilitate ring opening. And now when you get your ring closure, you have a 6-member ring here where because of this group, it's polarized. So now any elimination, any attack on that carbocation is regiospecific. So all the reaction is going to happen here to give you your elimination or trapping with the presence of a nucleophile. So that's the baseline premise of the reaction we were working with, and that's the baseline which we started all our reaction development. So what I'm going to do now is I'm going to highlight some of the previous work we did and start into the first vignette on a natural product target that we're trying to finalize right now with the purposes of doing some structure-activity relationships. Okay. So in the first example, we're going to talk about this appendage manipulation-based DOS. And so what we're looking at is taking that cyclopropane, and what we did was start with an alkenyl system, treat it with a Lewis acid, where we go through that oxyallocation intermediate. And what happens in this case is you get elimination, and there's 2 protons in the system that can eliminate your HA or HB to give you the alkene systems here. And so both these products are formed depending on the nature of the system, and they both are separable and isoluble. So you have an approach that can give you different systems that can be used to do further chemistry. But the important aspects are, like when you do the reaction, you get these 2 in this ratio, 3:2 is usually the setup for the system. But it works for, put a heteroatom there, it works. You can just have phenyl aryl groups, those kind of things, they work. This was really our initial foray into this and showed proof of concept. Ultimately, you can tune the reaction to give selective elimination, and you can do that using a silo group, where you basically take advantage of the fact that silo groups stabilize carbocations at the beta position. So if this is now silenced at a hydrogen, you get controlled elimination to give you selectively one product over the other. And you can do the same thing with the other isomer to get to that product itself. Okay, so this was with the alkenyl. This was our first publication in this foray, which was in 2010. This is one of the first examples of Lewis acid catalysis. It's a fairly broad substrate scope for the system, but this was really where we wanted to go, was really do this with aromatics and heteroaromatic systems as the pi unit. Because what that'll give us is fused cyclohexane rings to benzene rings to heteroaromatics, where there's utility in that, because when you look at things like the Diels-Alder reaction for the formation of 6-member rings, often for fused systems, those reactions aren't as efficient, or you require certain specific substrates that you have to make to be able to accommodate those different reactions. Okay. And so the next foray was into the heteroaryl groups, where now we have a heteroaryl unit as the pi system. Indium triflate is our catalyst of choice. We're using 5 mole % of the catalyst. I'm showing here that it potentially can work for different substituted heteroaromatic groups. How do we make these systems? We start with a beta ketone ester. We do a diazo transfer to get to alpha diazo compound. And then what we're doing is a rhodium catalyzed cyclopropanation, where we make a rhodium carbene and then do the cyclopropanation. Okay, and that's true for whether it's a 2-substituted or 3-substituted heteroaromatic system. This is the rhodium catalyst we use. We're using these dirhodium catalysts. This one in particular was developed by Justin Dubois at Stanford. But from these systems, we have the cyclopropanes, then we can do the cyclization reactions. And for this system, it works pretty well, whether it be for various heteroaromatics. And so this is the baseline reaction, which we will take into discussion for the actual total synthesis reaction. Okay. So from here, the next thing we're interested in doing is doing a total synthesis of structures that have this kind of framework. And so there was one specific example of the compound, is Propolisbenzofuran B, which was isolated in 2000. Fairly simple molecule. Looks like it would be very easy to put together and make. It was isolated from Brazilian propolis, which is bee glue. If you're not familiar with propolis, it's basically bees use it to maintain sterile environments within the hive, prevent microbial growth, things like that. And it's been used in folk medicine, and you can buy bee propolis powder and things like that at GNC and all those other places. But they've been used in folk medicine for a long time. But they're like over 160 different compounds that make up bee propolis. And specifically, this was one compound that was isolated in 2000. It was shown to have some cytotoxicity against murine colon carcinoma cells and human fibrosarcoma cells. Modest activity, not the greatest, but we were interested for two reasons. One, that framework fits in with the methodology that we're developing. And two, if we can develop a efficient method, then we can also develop a library that would allow us to do some structure-activity relationships. And so we're looking at doing the diverted total synthesis. So Propolisbenzofuran has a benzofuran fused to a cyclohexanone ring. It's unique in natural products. The first total synthesis was published in 2014, and that was 17 steps. I'm being generous in saying 3.3% overall yield is really closer to in that range of 1.5 to 2, depending on how you actually do your calculation. The main drawbacks of the synthesis, they're linear, so you have no opportunity for your diverted total synthesis or diversity origin synthesis, and there's very low overall yield. There's a lot of use of protection and deprotection steps, and there's a number of inefficient or low yielding steps for the process. Okay, so we envisioned a process where we can actually generate this molecule, but in a modular way that allows us to make derivatives, make analogs pretty readily. So the way we envision this synthesis, so this is our retrosynthetic analysis. So starting from here, this Propolisbenzofuran would come from this precursor, where basically what we've done is just manipulating functional groups. So here we have the ester, so we have beta-keto ester. We're moving this ester group. We're doing deprotection, oxidation chemistry to get to the target. This compound here originates from our ring opening cyclization chemistry that I discussed before. So that's the chemistry that comes from here, similar to this, where if you look at this is with a benzofuran, works well, same general idea, same kind of chemistry we're looking to use for the system. Okay. This would originate from the cyclopropane, where we envision a modular approach where we would take this bicyclic cyclopropane derivative, treat it with a functionalized benzofuran, where we pop open this ring to access this core structure here. And what this allows for is various modularity, in that we can synthesize derivatives of this piece, we can synthesize derivatives of this piece, bring them together, which means we can functionalize the full periphery of the molecule. This cyclopropane basically comes from an intramolecular cyclopropanation from this diazo compound, which originates from ferulic alcohol, which is a commercially available natural product, which you just cap as the isopropyl, where the natural product has a OH here. Okay, so we're starting with a natural product, going to acylation, do diazo transfer, so on and so forth. And so we'll work through that, the actual synthesis, as we're working through this method right now. So we started with a model system just to show proof of concept. So starting with this cinnamic acid, we do esterification, then reduction of the carbonyl to give you this allylic alcohol. Acylation with commercially available methylmalonyl chloride gives us to this mixed malonate derivative. Treatment with tosyl azide, you guys do diazo transfer to get to your desired cyclopropane precursor, which then when you treat this with your rhodium catalyst, you get to your bicyclic compound. And so from here, we just wanted to do proof of concept. So we just treated with benzofuran and just lithiated benzofuran, treated it with this system to get to this precursor, where we just capped it as the acetate, where if you look at the natural product, it has the acetate in the natural product. So we're pretty much where we want to be in the system. And then you just treat this with Lewis acid, and you get the desired cyclization to occur. Happens in high yield. And what we see when we do the model system is we observe a J value for the coupling between these protons of 10.3 hertz. You compare that to the literature value of 9.9, so we're pretty confident, along with the other data that we have the desired stereochemistry for the actual natural product. And so we've done this in a way that's fairly modular with the two pieces, and so now we're working on the actual total synthesis using the real precursors. Okay, and so if we go back to the retrosynthetic analysis, now you're looking at how do we build this functionalized precursor, and then how do we build in this functionalized precursor as well. So to build it, So we actually started with vanillin just because we had vanillin in the lab. We didn't have ferulic acid ready to go. So we just started with vanillin. We protected it and then did a olefination reaction to get to the ester. Then we did the same process as you saw in the last previous slide, where you do reduction of the ester to give you your allylic alcohol, do your acylation, diazotransfer, and then cyclopropionation. So now we get to the cyclopropane that has the actual functionalization that you need in the actual target molecule. Okay, so modular approach. So now if we wanted to basically get to a different substrate, we just could easily change the different components of the system to get different functionality. Okay, so that's for the bicyclic system. We're currently, we're currently working through preparing the benzofuran. We're doing 2 different methods. We had started on this approach here based on some known literature. Unfortunately, we've been having some trouble with that. The scale that they've been doing in the literature is usually upwards of 20 to 30 grams, and we've been doing it on anywhere from 200 to 500 milligrams. So we have to optimize this approach to get to the desired product in a high enough yield. But in the meantime, we're doing a more stepwise approach to get there so we can finish the total synthesis, where we do start with a simpler precursor, do the cyclization to get to a functionalized benzofuran, and then do some other chemistry to actually build in that system up front. And so there's some other chemistry that's been established, and we're getting close here. Only a couple more steps to do this before we're ready to actually finish up the synthesis. Okay. But nonetheless, you know, the approach is fairly modular, allows us a lot of flexibility and being able to think about, start making derivatives. So the next set of studies we're trying to do with this is, so here's your natural products. We're gonna actually want to make a lot of the natural products so we can start looking at what is the specific mode of action, because that is not actually known for this system. So we'll be doing some specific experiments where we collaborate to try and, you know, figure that out, where we may have to do some specific manipulation of the functional groups of this system. We also want to make a small library of analogs where we do everything from change one functional group at a time. So in this case, you remove the acetate. In this case, you remove the hydroxyl group. In this case, you remove the, no longer have the ester, you have the alcohol, and so on and so forth. And we want to be able to do this and then actually get biological data for each of these systems and compare. And hopefully what we're starting to see is we can predict, based on these structure activity relationships, which systems will actually give us higher potency. You know, as a starting point, we'll have to worry about other factors later, but start with potency, but then also look at mode of action, because once we can figure out mode of action, we can get a better model of how can we predict selectivity. Is it something that's actually going to be therapeutically viable or not? That's the next step of this project. That's where we're looking to take this. So for this part, summary, again, our proposed sequence is really about, I would say, 10 to 12 steps, and that's giving some flexibility for if we have to do some extra protection and things like that, compared to the 17 steps that's been previously synthesized. It also, this approach is modular, so we just basically have 2 main components where you can bring them together. And there's a lot of opportunity for asymmetric synthesis if you want to do the, make the enantiomerically pure systems, because particularly what we'd have to do is just focus on this step here, where we do this with a chiral rhodium species, and you get a chiral cyclopropane, such that when you react this with the benzofuran, your resulting system will be asymmetric, will be chiral. And then you do the chemistry, and you should carry through that to the end. Okay, so that was that first part. And then in the next part, we're going to focus on the substrate-based dots where we're altering the pi system. And one of the things that we're looking at is, instead of having a carbon, you know, connected pi system, we have a nitrogen connected system with an active pi, you know, nucleophile off the nitrogen. Okay, and so the inspiration for this is based on the fact that we know from our previous work that you can take an indole, react it with under the conditions, and get to a desired ring closure. But what happens if you take the structural isomer, where now it's an acylated system? In both cases, the 2 position is free and reactive. So in this case, if that reacts, instead you get ring opening, you get attacked by that system, you end up with this type of core structure. This is your Hydropyrido indole core structure. That's present in a number of target molecules. So this is where we start thinking about the modularity of the method. We start thinking about, okay, I can switch out the different indoles and start accessing different types of compounds for the overall reactivity. We built a method that is pretty robust to do this chemistry, where you basically start with an indole, do an acylation with methyl malonyl chloride, and you can do this with no base, just heating. We can do this upwards of 30 to 50 grams. You get pretty much quantitative yield to get to the beta amido ester. Do diazotransfer to get your alpha diazo compound, and then do cyclopropanation to give you your active cyclopropane. And all these steps are pretty much greater than 85% yield, upwards of quantitative depending on the system. And then if you treat it with a Lewis acid, we pretty much get quantitative conversion to the desired 6-member ring. Okay, and so this is the kind of method that we're inspired by. It's pretty robust. It works for a wide range of substrates. I'm not going to go through the total substrate list here, but it works for a wide range of substrates. And it allows us to start thinking about things in a modular fashion based on, can I change out the indole? Can I change out the alkene that's used to make the cyclopropane? Because that allows us to basically change all the different components of the molecule. Okay. We actually took the method to make the system and built it into a continuous flow process. We published that in 2015, showing that we can do this on a large scale, upwards of 5 grams per hour in a continuous flow setup. But that leads us into the next vignette that I want to talk about, which is going after the synthesis of Rhazinicine. Rhazinicine was isolated in 1999 and again in 2001 from this Malaysian Kapsa plant. So this is your Kapsa plant here, and it's primarily located in Southeast Asia here. It has strong cytotoxic effects against your HeLa cell line, so that's the human cervical cancer cell line, with about 2.9 micrograms per mil, it's pretty cytotoxic. The problem is it's also cytotoxic against normal cells, so it's about 20 micrograms per mil. So one of the things we're interested in is, can we develop a modular approach to making analogs that allows us to maintain cytotoxicity or increase cytotoxicity while decreasing cytotoxicity for human cells. So that's the plan, the long-term plan for this project. This compound is a member of a larger family, where here's just some examples where you have this large ring system, this large lactam, but you have different oxygenation on the ring system, whether it be directly on the parole or the parole itself is oxygenated. And so we're really interested in targeting this compound because it fit right in with some of the methods that we were looking at. Particularly for Rhazinicine, there have only been 2 syntheses, while some of the other compounds have been synthesized in great depth. Both of these, so 11 steps, no overall yield reported, 14 steps, no overall yield. We're looking at a method that would take us ideally less than 10 steps to be able to do the chemistry, but in a modular fashion. And so earlier last year, we developed a general method where we can start with the parole. So I showed you where we had the indole, same kind of setup, and now we just switched out the indole for parole to do the cyclization. Different catalysts works well for this, but be able to do that to get to these tetrahydroindolizines. Again, works for a range of substrates. So basically just changing, again, this would be from the alkene, changing different alkene units to get to different structures. Then to actually generate the target, we envision doing this with this type of ester, doing the cyclization, and then basically building the functionalization at the end and creating this bond in the last steps to generate that macrocycle. Okay, so that's how we envision it. Modular in the sense where you can build this, bring in another unit where this is the amide you're forming, and then bring in the cyclization at the end. So you have a lot of room for derivatization and manipulation. So just to highlight where we are now, you start with this diazo compound, which we can generate in 3 steps from commercial material. So you take this, you can make this ester in basically 2 steps. So cyclopropanation between this ester and that diazo compound gives you this cyclopropane. Treat it with the Lewis acid, you get your cyclization to occur readily. So we can get to here in 65% yield. Get rid of the ester group by doing this decarboxylation approach. Saponify the ester with sodium hydroxide, followed by amidation with an aniline derivative. So iodoaniline gets you to this compound here in 70% yield. And then what we're doing right now is working out the conditions to do this cross-coupling, the CH functionalization between this carbon and that carbon. And so there are some conditions that have been established, but they all involve having a protecting group on the nitrogen. And so what we're trying to work out is doing this without the protecting group to be able to do this directly, because that will save a step, keep it at 9 steps for the synthesis. But if we can access either this system here or even get to the precursors, we can start accessing some of the members of the family of the family of these compounds. So for example, if you take that precursor here, basically do a deoxygenation of the system, and then do your cross-coupling, you get to Rhazinicine, which is one of the members of the family. If you take that and do a pyrrole oxidation, you'll get to gluconolam, which is another member. So you're going from 9 to 10 steps, 11 steps for this one. Similarly, you can access the other members by starting with a different pyrrole unit where it's functionalized in this way. Do the same chemistry leading up to the, building that complexity, deoxygenation, cross-coupling, and now you get into all the different members of the family. This followed by oxygenation gives you that system. So simple method, but then it's modular enough to access all the different members of the family, which then we can look at for biological activity, start making derivatives to see if any of them will be more potent than the parent compound. And these are all interesting because they're all antimitotic compounds. They have a lot of potential, but they're all fairly cytotoxic. So if you can tune that somewhat, we might be able to get into an area that is more therapeutically relevant. Okay. And so just to summarize this part, we're looking at a 9 to 10 step proposed synthetic sequence, modular, so rapid synthesis of cogeners, in no more than 12 steps. And again, your Homo-Nazarov cyclization and late stage CH functionalization. Given the time, I think that's a good place if, based on the time frame of the class, is a good place to stop. I just wanted to highlight 2 vignettes where we talk about synthesis, talk about being able to access compounds in a rapid fashion, so then we can start thinking about derivatization and so on and so forth. Just to highlight one other thing, in terms of modularity, our methods will allow us to access compounds in such a way that we can start, I'm skipping on purpose. What I want to highlight is the fact that we can generate compounds of this type readily, where they're now all aromatized. And what that allows us to do is start looking at tunability for probes. And you can take these compounds, put them in cells, and start thinking about looking at these systems to see what they're actually interacting with. Can we tune the photophysical properties of these systems so we can, have them target various organelles, have them be fluorescent enough to be, either push that into the red region where you're pushing this above 600, things like that. So you have some tunability in the development of these types of derivatives, but all based on the same methodology that we're developing, where we're just changing different aspects in a mix and match type approach. With that, I'll stop, given the time frame. This was my group last summer, where we had some undergrads and RU students. So I always recommend, we have an RU program at Georgia Tech. It's a good place to, learn some good in-house skills and things like that regarding synthesis, biochemistry, PChem, whatever. And so all these students did a really good job in terms of creating an environment that was positive for those kind of experiences. But in the end, the ones in blue are the ones that really worked on this Homo-Nazarov type project. And I want to thank all those students specifically, collaborators for doing the various aspects of some of the other works, and then funding sources, particularly NSF, the ACS Green Chemistry Institute, and Oak Ridge for funding related to some of these projects. With that, I open up to any kind of questions or things like that.
My name is Michael.
I'd like to know, could you just review the process of how the substance is extracted from the bees?
Yeah, so for the propolis, so usually for the propolis, it's usually dried down, and then what you do is basically do a liquid extraction, usually methanol or some other organic solvent, and you basically do several levels of extraction to try to identify the different compounds in the process.
Hello, my name is Dylan Zimmerman. I noticed a fair number of the reagents used as catalysts could be considered harmful to human health. Does your lab work with exploring into using different catalysts? And if so, do you notice any difference between using these catalysts versus the more safe ones?
Yeah, so some of the catalysts that we use, particularly because on an academic scale, we haven't been as concerned, but as we start to get to the more idea of scale up and get into the ones that are more applicable for industry, we have been starting to collaborate with a group that does solid supported catalysis, where we're looking at, for example, like the rhodium catalysis. Rhodium is fairly expensive, so looking at putting that on a solid support so we can recycle the catalyst and reuse it. We also are looking at utilizing more main group catalysis. So we've actually been doing a lot of work with calcium catalysis and things like that, trying to move away from some of the more harsh tin and some of the other reagents like that. But there are a number of groups at Georgia Tech that are looking at various catalysis, everything from heterogeneous catalysis to other types as well.
Hi, my name is Kadra, and I was wondering if you know what properties about the Rhazinicine make it such a good medical property for the body.
Mm-hmm. So it's a tubulin binder. So based on the nature of the structure, it actually binds tubulin very well. It mimics what you see with Taxol. And so that's part of what's so interesting about it, because it does that, but it also behaves slightly differently. And so that's part of what is trying to figure out really what the full mode of action is, because it has similar properties, but it actually does some other things as well.
Hi, my name is Obed. I was wondering about your propolis. And what was the yield that you guys got, and what kind of yield are you expecting to get after you optimize that reaction?
Right. So ideally, we're hoping that after we optimize, I'm hoping that the overall synthesis, if we're sticking within 10 to 12 steps, I'm hoping that we can get it within 15 to 20%. That's my target goal. Doesn't seem like a whole lot, but when you're 10-step synthesis, if we can do that on a scale up where you can then generate a lot of analogs, I think that's fairly reasonable to be able to then translate that ultimately, if we do find something that's really interesting, to potentially go towards company or industry, or where we can do more industrial scale.
Hello, this is Dylan again. I noticed on one of your first slides you had what looked like a dendrimer. Could you talk a little bit more about what your group has done with whether it's that particular dendrimer or any dendrimers?
So yeah, so this structure here. Yeah, so what we're interested in doing is one of the methods that we're trying to develop in my lab is where utilizing basically almost like a click-like reaction where we can generate, say, one of these units in rapid succession. So we take advantage of cyclopropane ring cyclization chemistry. So whether we have 4 thiophenes or some other conjugated system, and basically click on a diazo unit, gives a furan or benzofuran or other system like that, and we can build those dendrimers up very rapidly using this click-like chemistry, which would avoid having to use palladium to do the cross couplings. So we haven't published anything yet with that. We're building the methodology to do those kind of things.
Okay, well, we want to thank you very much again for your presentation. Very clear and very powerful method that you presented. And just a reminder, some of our students may be interested in doing some summer research. If you could send me an email, I'll then send it out to them.
Yes.
Get more information.
Yes, we'll do.
Thank you again.
Thank you again for the invitation.
All right.
All right.